Okay, good morning, everyone. So this is Grand Rounds today. So it's a new session. We had a very special guest today, Dr. Kahle. He is a neurosurgeon at Mass General. I can say a couple of things. He's a pediatric neurosurgeon, and he has like multiple cases. They can see multiple conditions, but the other part that he has a very strong research and active research. So that's very something special, and I want to just bring all of this to us to give us some insights about these like congenital neurological diseases. That's the main thing. I want to just like welcome Dr. Kahle. Just thank you for like giving us this special time from your very busy schedule and share with us all your like work. So I give you all this spotlight, and we can start now.
All right, I really appreciate it, guys. Thanks for the invitation. It's a privilege to be able to communicate some of our work. Today, I'll be focusing mainly on the congenital hydrocephalus project, but we'll also introduce a platform that we've been able to apply to several other congenital neurosurgical diseases. And so I'll just get right into it. I have no financial conflicts of interest, and I wanted to express gratitude for our funding sources, which makes the work possible. So I'm a pediatric neurosurgeon and an investigator, and neurosurgery and neurology are fields that are predicated on precision. Some of the major leaps in our field have been driven by advances and innovations in technology that have allowed increased resolution to happen, whether it's with the introduction of the neurosurgical microscope in the 1970s, brain lab neuronavigation in the late 90s, and some other, technological advances that allow increased precision and accuracy to happen in the operating room. I think it's a challenge of probably my generation and the next generations to increase that resolution down to the molecular level. And of course, this is happening already in certain subfields within neurosurgery, within neurology, like neuro-oncology is probably a classic example. But it hasn't happened to the extent in neurodevelopmental disorders that are treated by neurosurgeons. And so the question is, can we increase the resolution that we're looking at these diseases with? So as you guys well know, neurosurgeons do some pretty crazy things. And here, just several examples of some of the things that we do and actually still do. So they're all for life, saving purposes, and they do save or promote life, but they're still pretty radical and pretty rudimentary if you think about it. So for addressing diffuse cerebral edema, whether it's from stroke or anoxic brain injury, we do a decompressive hemicraniectomy, simply removing half the skull. For the treatment of CSF accumulation in the setting of clinical hydrocephalus, we stick a catheter in the ventricular space and drain fluid. And this is an example on the right side of a anatomic hemispherectomy. These days, we're doing a little bit more functional hemispherectomies, but we still lop off major parts of the brain in an attempt to produce a therapeutic effect. So, while these maneuvers are all life-saving or can be life-saving, I think the bar is set pretty low for some of our interventions, and there is room for improvement. So this is not meant to be interpretable, just the opposite. It's a famous wiring diagram drawn by famous American physiologist, Dr. Guyton, in the 1970s, where he was trying to depict the overlapping networks. One of the first examples of systems biology in which he was trying to convey the regulatory networks for blood pressure regulation. And you can see there's influences of the kidney and vasculature, the brain. And so the question is whenever you have a complex homeostatic system such as this, what are the key regulatory nodes within that network that you can pinpoint accurately to influence system behavior as a whole? And so if you're interested in something complex like CSF dynamics, production, transport, reabsorption, what are the critical nodes in this labyrinth that you could target to influence, system behavior in a therapeutic way? And I would posit that genetics is a agnostic approach that allows you to do just that, down to the gene level, and then even beyond that, down to the level of single amino acids, where you can, by mutating a single residue, have a profound effect on system behavior. So that's the approach that we've taken that plays to my strength as someone trained in molecular genetics with Rick Lifton at Yale in the MD PhD program. It is an unbiased agnostic approach that lets the biology and not your hypothesis tell you where to go and what the next steps are in the investigative pathway. It is able to define key components of complex pathways down to DNA-based pair resolution. And as Rick would say, you're dealing with the human model system, in which by the fact that you're starting with human disease processes in humans, that the findings are going to be relevant. From this, you can gain insights not only into disease pathogenesis, but also into the genetic regulation of normal development. And this is a tried and true platform and approach that, by studying even rare forms of disease, you can uncover pathways that can be targeted for more general purposes. So, for example, HMG-CoA reductase, mutated in a handful of patients in the world, is the target of statins, which I currently take in the morning. And PCSK9, again, discovered in only a few families, but now being targeted and given to millions of patients. So our hypothesis is that we can increase neurosurgical precision using an integrative genomics approach, and specifically apply to diseases that we treat in pediatric neurosurgery, that for historical reasons, right? Usually neurosurgeons aren't conducting major and perhaps rigorous investigations. Neurologists are, and because there's career pathways that support that kind of physician scientist platform. Whereas from a neurosurgery standpoint, at least historically, there's been some very cool changes the last 5 years or so. It's far from what the neurologists do, or at least the time that's allowed and dedicated. And there's good reasons for that too. But by applying an approach like this, we might be able to address some of the unmet needs for patients. Many of our diseases that we handle, both in pediatric neurosurgery, but also pediatric neurology, are riddled by relatively antiquated diagnostic classification schemes. So even just saying something, hydrocephalus, or what does that mean? There's Chiari-1 malformation. There's anybody who does any sort of clinical medicine knows that there's a myriad number of clinical subtypes that have real impact on treatments, prognosis, the way that you communicate the potential utility of offering a surgery to a patient. It really depends on those different subtypes and getting more granular about what a particular disease is. Addressing the needs that I just mentioned would have obvious impact and benefit by increasing precision for family counseling, prognostication, choosing the right surgical treatment or drug treatment, And then, especially from a pediatric neurosurgery point of view, pediatric neurology does it way better. But in pediatric neurosurgery, and remember, a lot of these patients are kind of, for good or for bad, owned by us or within our domain. We have a pretty low bar for what we call success. And so, how many times have the pediatric neurologist reached out to the neurosurgeon inquiring about whether or not this could be a shunt problem, and the neurosurgeon comes by, taps the shunt. Yep, it's working. Nothing more needed to do. We've done all we can from a neurosurgery point of view for this, for this child, which essentially is, maintaining survival. So I would say, instead of how we define neurosurgical success, moving beyond that definition to one in which we're trying to actively optimize neurodevelopmental outcomes, which in some instances may be withholding a surgery or choosing a different surgery or doing it at a different time point. Because there's trainees in the audience, before I get into the meat of the scientific talk, what I wanted to do is to just deliver to you the concept of vocation, which I know you all know, but this is a particularly good quote that allowed me to, at least in my own path, think about what to focus on in my career. And this is from the Methodist preacher Frederick Buechner, who said, "Vocation is the place where your deep gladness meets the world's great need." So as you go forward, especially as potential physician scientists, this is a difficult path, and it's it's difficult for anybody, whether you're a neurologist or a neurosurgeon. But in neurosurgery, I think it's particularly difficult because there are, the length of training, the rigor needed for technical skill in the OR. I think the difficulty with competing against other people who may be doing it completely full-time, without any clinical obligation. But I think that there is tremendous area, there's a tremendous opportunity, both for neurosurgeon scientists and people in pediatric neurology, to make huge contributions in these in some of these diseases, because to put together these cohorts, to structure studies, you need to know human disease, how to recruit patients, access to patients, and inspiration about tackling clinically relevant problems. So when you're beginning to think about your own particular path, I would just ask you to think about, what problem are you particularly passionate about? And if you solve it, will it help somebody? You can be passionate about a lot of different things, but if you solve that problem that you're passionate about and it doesn't help anybody, that might be a problem. On the flip side, if you are pouring out your blood, sweat, and tears and helping people, or addressing a particular need, but you're not passionate about it, your cup is going to run dry, and it's not a sustainable venture. So for me, being a neurosurgeon scientist, it also helps put distractions and frustrations in perspective. For me, there's few things more exhilarating than making a discovery in the lab. But anybody who's done any sort of time in the lab, there's nothing more depressing when things aren't going right, and that's usually the state of affairs in the lab. It's like a bit like being a hitter in the major leagues, where if you're hitting 280 or 300, that means you're failing the majority of the time. And so it's all about persistence, and that persistence is easier when you're addressing a problem that you're passionate about and it's helping somebody. For a neurosurgeon, what can we uniquely bring to the table in these paradigms? I mean, the idea of making the OR an extension of your lab is a great one. Our functional neurosurgeons do a great job at this. Recording different outputs from the brain in awake patients during procedures in the operating room. from a pediatric neurosurgery standpoint, there's really unlimited access to tissue, and also having a pretty good knowledge, like you guys, about brain anatomy, whether it's from an imaging basis or from a gross anatomy perspective. And so developing a program that plays to your clinical strength, I think, is also important, in which your investigations or discoveries for diseases that you treat can illumine, clinical pathways for treatment, and bring more patients to you, which not only augments the research that you're doing, but inspires, new questions for, to answer. One other concept is, or really term, is that of kairos, which is, for the Greek, anybody who knows Greek in the audience, or is a philosopher or a theologian, they might know that this Greek term for time is different from the chronological term for time, chronos, like, minutes to hours. Kairos is more about the idea of an opportune moment, or timing for decisive action. And so as you embark on that early stage in your career, which frankly, I feel, which I'm still in right now, so I say this with some hesitation, but it's about, especially when you land your first job and your feet are on the ground, you say, given the confluence of technology, where I am, my environment, the support that I have from my mentors, my particular interests and past training, what might I be ideally suited for at this moment in time? And it may not be exactly what you had planned, maybe years before. So from an operative standpoint, I love, P-fossa tumors. They're, from a technical standpoint, from a interaction with families, these are some of the most fun clinical situations that I'm involved in. But depending on where you are and what's been done in the field, being a cancer biologist at this moment in time for those type of tumors doesn't make sense. The field's too saturated. A lot of the key low-hanging fruit findings have been discovered. It's highly competitive, and you have to say, how could I make a unique contribution at this moment in that environment? In contrast, you look around you and you say, wow, there's a ton of these patients with congenital hydro or arachnoid cyst, Chiari-1. No, there's much less has been done, and they happen to be coming and knocking down my door to be treated. And you realize that they're sometimes inadequately treated because they're perhaps a neglected patient population, or it's a less sexy problem that hasn't been historically earmarked for funding, like brain tumors, for example. So you might have to rebrand yourself if that's what's necessary to take advantage of your environment and to answer an important question that can still help somebody. So we've taken this approach with some success that has been, I would say, largely dictated by the environments that I've been in, and the students working with me. These are just a couple examples of people who have from a range of postdoc like Peter Jin on the bottom to MD PhD student Zui Fan in the middle, to now Barrow Neurosurgical residency fourth year resident Shruta Fury, who actually just had a baby a few months ago, to Jason in the upper right-hand corner, who is applying for neurosurgical residency and interviewing as we speak. So we've taken this approach and applied it to pediatric neurosurgical and neurological conditions. So the one that I'll be focusing on today, I could pick a couple of different ones that are in the lab right now, different projects, but I'll focus on the Hydrocephalus project. Actually focus on the congenital hydrocephalus project, so not acquired. That's another kind of line of investigation in the lab. And the reason for doing that is because I think it's a kind of a prototype pediatric neurosurgical neurological disease. And the project is probably the most mature in the lab just because we started it first. So you guys, everyone on this audience knows about hydrocephalus, but I'll remind you that it's common and that it's treated by largely by CSF shunting or endoscopic approaches. So these are approaches that at least in conceptually have really not changed in decades. Some of the first shunts were placed in the 1950s. They've saved thousands of lives, and they can be life-saving. But from a definition standpoint, which influences whether someone or not gets an endoscopic treatment or a shunt treatment, we really rely on a functional definition of hydrocephalus that is one between communicating and or synonymous with non-obstructive versus obstructive. If something is obstructive, for example, due to aqueductal stenosis in the lower right-hand corner, you can choose to apply something called an endoscopic third ventriculostomy, which provides an alternative conduit and exploits the endogenous CSF reabsorption pathways in the brain. Whereas in non-obstructive pathology, which is a ton of cases, by the way, more so than aqueductal stenosis, you really rely on shunting, which is, again, life-saving, but it's the #1 medical device failure, cause of medical device failure in the United States. It's not accessible to many other parts of the world where neurosurgeons and technology, shunt technology is limiting. And, after 2 years, about 45% of these things get either infected, obstructed, and either need to be replaced, revised, and so forth. So this is a major problem, and we could do better for treating patients. I'll get into this in a little bit, but a key concept that I'd like for you to take away from this is that not only are we interested in finding alternatives to shunting, but the idea of shunting itself in certain contexts, I will posit is negatively impacting health, because I think we're choosing to do this sometimes in inappropriate situations. And I'll give you a couple key examples. But also just a brief reminder that, and everyone knows this who I'm talking to, but there are primary forms of hydrocephalus, also known as congenital, developmental, now genetic, and then acquired forms, which are secondary, a result from a known secondary cause, such as hemorrhage and prematurity from germinal matrix hemorrhage, infection, meningitis, or other types of CNS infection, and of course, obstructive neoplasms. So there are multiple obstacles to gaining additional information about the pathophysiology of hydrocephalus that might open doors to improved understanding of CSF dynamics, coming up with new therapies for hydrocephalus. And that in part is due to our limited knowledge of the genetics of this disorder. So while greater than 60% of all congenital cases have been predicted to have a genetic etiology, until a couple of years ago, mutations in known genes only accounted for less than 5% of these. There's reasons for this. Some of the traditional genetic approaches that capitalize on large families with multiple affecteds that lend themselves to more classical genetic approaches. For congenital hydrocephalus, there's not too many of those, and of course those sorts of families were the source of the genes that have been discovered so far, like L1-CAM, which I know you guys know as being the most common X-linked form of hydrocephalus, often associated with aqueductal stenosis, but also part of L1 syndrome where hydrocephalus may not be present. That's a key finding, and I'll talk about that later. There's also some non-syndromic autosomal recessive forms of hydrocephalus. MPDZ was discovered in a highly consanguineous population in the Middle East that lends itself to human genetic discovery by offering rare large effect recessive alleles. But in the majority of hydrocephalus, it's sporadic. So greater than 95% of cases are sporadic. So the question is given the locus heterogeneity, phenotypic complexity, and the fact that the majority of cases are sporadic in nature, how are you going to apply genetics in that in that context? And really starting in 2009. With major advances coming out of Yale and Seattle, Johns Hopkins, and some other groups, it's become clear that whole exome sequencing and then more recently whole genome sequencing offer pathways to discovery of rare large effect gene variants that would otherwise have gone undiscovered. So we set up something called the Hydrocephalus platform, whose objective was to utilize an integrative genomics approach to discover, validate, and then gain mechanistic insight into the newly identified genes underlying congenital hydrocephalus. Its aims were threefold and very straightforward. One was to recruit, expand, and then characterize the largest congenital hydrocephalus cohort in the world. And to do that, there were a few things that helped this process. One was to not only employ traditional physician-directed referrals, but also collaborations with industry partners who have a lot of exomes and good phenotypic information, along with family-centered social media engagement, which increased our reach and certainly increases the efficiency with which you can identify and engage families who might have a little one with hydrocephalus. Technical advances in artificial intelligence, machine learning have changed the game for allowing us to phenotype our patients, both by scanning, the medical record and looking for HPO terms by AI programs, or by just examining hundreds and sometimes thousands of brain MRIs looking for patterns. This allows one to make genotype-phenotype correlations and find associated endophenotypes that are associated with different genetic variants and different congenital hydrocephalus subgroups that have real clinical value. So big cohort and deeply phenotyped cohort, aim 1. Aim 2 is to use whole exome sequencing, genome sequencing, both for the germline and capitalizing on the fact that many of these patients are our own in our group. We are able to get tissue from the OR for somatic mutation or variant detection in actual brain tissue by having paired germline and brain samples. So for the majority of these studies, we're most interested in looking for rare large effect de novo mutations. So mutations that are not transmitted from mom or dad, but occur spontaneously after egg fertilization, during cell division, and which account for a huge percentage of the sporadic cases that we see, and I'll get to that in a second. So beyond just gene variant detection, with the increase in information about gene expression profiles, both from bulk RNA-seq and now single-cell RNA-seq, we're able to integrate our genetic findings with other multi-omic data to define how these gene variants might be impacting brain biology in both time and space. So looking for different epochs in brain development where genetic variants may converge in certain pathways to impact and ultimately lead to the brain phenotype that we're interested in this case, congenital hydro. And last, when you're dealing with variant detection, there are many cases, when you're finding an extremely rare or novel mutation and it's a recurrent mutation only in diseased individuals and absent from control cohorts, making a case for pathogenicity and causality of that variant are not very difficult. But in other cases, in some of our rare damaging transmitted variants that are clearly incompletely penetrant or yield phenotypic heterogeneity, even within the same family, validating and being able to study high confidence CH genes in vivo not only can help you make a case that this is in fact a bona fide disease gene, but you can also get information about how that gene is impacting normal brain development and is impacting the pathophysiology of disease. So to do that, we really use Frog as it's definitely not a high throughput. I would call it a medium throughput platform for gene validation, and mouse to focus on those genes that are particularly interesting to us, whether because they're highly mutated with high frequency and therefore could impact a lot of people, or if we just think it's a cool gene that's impacting, that has the potential to impact interesting biology. So the significance of setting up and performing this endeavor, this research endeavor for congenital hydrocephalus will be, remain to be determined by posterity here. But already we see that it's been able to provide some unique insights into human brain development and function. We've certainly been able to uncover pathogenic drivers of disease that are responsible for almost a quarter of congenital hydrocephalus. We've been starting to define congenital hydrocephalus subphenotypes that are helping us improve prognostication and treatment decisions in regards to whether or not we place a shunt or whether or not we would consider an endoscopic treatment. And then we hope that these findings by us and others, we're not the only group doing this, of course, encourage the development of precision medicine therapies, which is being done already mainly in neurology for multiple disorders, whether it's for spinal muscular atrophy or other Mendelian conditions for which, whether it's gene replacement or base editing out a missense mutation could effectively correct the disease. So these are just some examples of our reach. This is for domestically, but we still recruit patients from Canada or south of the border, certainly across the Atlantic into Europe through social media. And then of course we have physician collaborators at Boston Children's. I'm at Boston Children's now in part along at MGH, along with MGH, but with CHOP down at Johns Hopkins and a bunch of other people that are involved. And so this is just a kind of a cool example of using UMAP clustering to illustrate how, for whether arachnoid cyst, congenital hydrocephalus, we're uncovering certain themes where we are able to map certain gene variants to certain disease subgroups that are defined by HPO terms, human phenotype ontology terms that cluster very tightly, actually, that you would otherwise really not notice otherwise, by the fact that some of these associated endophenotypes like hypotonia or seizures are either glossed over in the medical record or not tightly tethered to the specific diagnosis in that patient. So we're able to define subclasses of disease that have prognostic value. I'm going to go through the published study here from 2020, but we'll be having one that has about 5 times the number of trios that we've been able, 5 times the number of trios that were reported in this paper that should, be coming out pretty soon. But I'll maybe we'll round back after that study is out, and we can talk about that and other future things. But this was a study in which we recruited 381 patients with shunted hydrocephalus. So this was not just at our institution, but really a great cross-sectional study of patients from multiple institutions that were shunted by a neurosurgeon with congenital hydrocephalus. And we studied controls alongside that. So we have a pretty robust computational pipeline now for the analysis of both de novo and inherited variants that looks for high confident rare single nucleotide variants and small indels, deleterious missense mutations, loss of function mutations. And we used statistics to find and compare whether or not these variants are being enriched in cases versus controls. We do one-tailed binomial tests to look for enrichment of those variants. And then we also look for their presence or potential enrichment in control populations. And it was interesting that we were able to pick up known familial congenital hydrocephalus genes in about 2.1% of our sporadic congenital hydrocephalus cases. These are all patients that we recruited without a genetic diagnosis when they got involved with the study. So even just by applying whole exome sequencing to a patient that comes in through your door with a new diagnosis of congenital hydrocephalus, someone who's been shunted, about 2% of those cases are going to have one of the known genes. And then beyond that, we were able to find that damaging de novo mutations account for nearly, now we know from our increased cohort, about 23% of cases. In this particular study, it was almost 18%. But the majority of these de novo mutations are impacting genes that are highly intolerant to mutation in the genome. So with PLI scores greater than 0.9, these are genes that are evolutionarily protected from mutation because of their importance, yet they're highly enriched in mutation in this phenotype. Beyond that, not only are they highly intolerant to mutation, but they're also highly expressed in the brain, as you might expect. So the overwhelming majority of them are expressed in the top quartile percentage of genes that are expressed in the brain. At this point in time, we continue to find TRIM71 mutation. All these are frequent flyers by now, but in this particular study, TRIM71 was the most highly mutated de novo mutation that we were finding in sporadic cases. Several of these approached or surpassed genome-wide significance for enrichment of mutations. And so in a relatively small cohort like this, it's great to have a medium throughput platform where you can knock down the gene ortholog in a system like the amphibian platform that we have. You can express the wild type human gene. You can then express the mutant human gene with the mutation that you've defined in the particular patient that you're studying. And you can essentially make a frog avatar of your own patients. And we do this, which is a bit weird, but you would not necessarily think that you can absolutely recapitulate ventriculomegaly and the development of aqueductal stenosis as seen on the right by engineering in a human-specific patient mutation in a frog depleted of the endogenous wild type gene. And so the patient on the left is a patient of mine with a CRB2 mutation, a missense mutation, and we knocked it down, and then we overexpressed the wild type, and we were able to see a wild type frog tadpole. And in contrast, when we express the mutant allele, the single base change, you see the development of aqueductal stenosis, obstructive aqueductal stenosis, and ventriculomegaly in a frog. A corollary to this is that in addition to recapitulating disease phenotypes, validating gene variants with a functional study, is that we understood and are beginning to learn that the ventricular system, whether it's in a frog or a mouse, is not the unidirectional flow system that one would usually think about, at least from the traditional, largely neurosurgical bulk flow model. Instead, there are subfields of CSF flow driven in part by cilia. But this is just an interest. I bring this up only to show that, by disrupting networks with disease genes, we not only can gain insight into disease biology, but also uncover normal aspects of CSF homeostasis, which are still incredibly cloudy. So here are some of the TRIM71 de novo mutations we've discovered in congenital hydrocephalus, all novel, and they recurrently hit a couple of residues in these so-called NHL binding domains on the C-terminus of the molecule, which are important for TRIM71's binding to its mRNA targets that catalyze the RNA target degradation. So our initial hypothesis was that this is something that is somehow impacting an evolutionarily conserved ancient pathway that people discovered in worms decades ago, important for stem cell regulation by the degradation of RNA targets, which when translated impact stem cell proliferation and differentiation. But this was not the first idea that we had in regard to what a hydrocephalus gene might look like. Our very simple-minded hypothesis was that maybe we would find some gain of function mutations in a choroid plexus ion transporter, localized at the apical membrane, involved in CSF production, or something impacting the development of the deep venous system, or the granulations that reabsorb CSF. So something in the secretory or reabsorptive apparatus that impacts CSF dynamics, but it was not the case. Nothing like that. In fact, all the genes that have been highly implicated in congenital hydrocephalus appear to be very important regulators of early neural stem cell development in the germinal matrix, or neuroepithelium in the case of TRIM71. So TRIM71 is through functional studies. Zwi's paper just got, not just, about a month ago, accepted in Nature Neurosciences. We're just beginning to learn the functional consequences of some of these mutations in vivo. But TRIM71 happens to be now a novel marker of neuroepithelial cells. It's one of the earliest markers of this very early neural stem cell population of the prenatal brain that lines the neural tube, with the primitive CSF being within that tube, and then very, very early on the developing brain ventricle. So Zwi has gone on to recapitulate congenital hydrocephalus in mice using a couple of different approaches. So one of these is just simply engineering in the orthologous patient mutation in the mouse gene of TRIM71, and this results in massive ventriculomegaly very early, even before even before birth. He's done prenatal MRIs in mice, which are very cool. He's been also able to recapitulate hydrocephalus by depleting the TRIM71 gene in a tissue-specific way or cell-specific way using two different Cree drivers that are specific for early neural stem cell populations, one in EMX1 Cree and another Nestin Cree. So in either one of these cases, conditional knockout of TRIM71 during the earliest parts of brain development, yet only in neural stem cell populations, causes and recapitulates the same hydrocephalus phenotype that you see with the point mutation. So giving insight not only to when this occurs extremely early, embryonic neurogenesis, during embryonic neurogenesis in utero, but then in addition to that, giving some idea about where this mutation is impacting disease biology. And so the neural stem cell population, particularly in the neuroepithelium. So mechanistically, impaired TRIM71 mutation leads to impaired prenatal neural stem cell regulation, and largely its impact is decreasing proliferation and then catalyzing precocious differentiation. So really what's happening is, and I think this is a central concept that I want to convey, is that and I'll kind of go backward to further flesh out the point. But what we're seeing when you look at this MRI is not massive ventricles due to progressive increase in intracranial pressure due to either increased CSF secretion or decreased reabsorption, but rather a very thinned cortical mantle that's a result of impaired neurogenesis starting in utero as a result of depletion of the neural stem cell pool due to a combination of decreased proliferation and precocious differentiation of neural stem cells. And so then to backtrack a little bit, mechanistically, how does this happen? These mutations in the NHL domain decrease binding to critical targets of TRIM71 that are important for neural stem cell proliferation and differentiation. Biophysically, what I think is very interesting is that, and maybe intuitive to some, but demonstrating it experimentally, I think is important, is that the hydrocephalic cortex, or I should say the very thinned cortex, hypoplastic cortex of TRIM71 mutant mice is highly compliant. And so there is no brain parenchyma to resist for any particular volume of CSF to counter expansion of that space because there's no tissue turgor. And so these are highly compliant brains. And despite that, they have normal ependymal cilia flow. They have normal CSF secretion rate. They have low to even normal to even low intracranial pressures. And the reason is that this is really not a disease of CSF accumulation per se. It's really a disease of a hypoplastic cortex. And so when you actually look through patients, and there are a number of different subgroups of congenital hydrocephalus of this type with scans like this and other scans, what you're seeing, and I think this is a critical point that has translational value, is that these patients who are being shunted, their ventricles do not get smaller. Their neurodevelopment does not improve. And therefore you have to ask, why are we shunting them? They may be macrocephalic in the 90 percentile, but if you look back at the notes of these patients, the operative notes, and again, these are coming from everywhere, and not just our practice or something, you're seeing that these the intracranial pressures measured are, in humans are 8 or 9, not 30, not 40. as you might expect with a little kid with a big head and ventricles of the size. Despite that, we are seeing how often these patients get operated on, and you have this own experience yourself, certainly on the wards, where a patient comes in like this. Maybe they've been shunted, maybe they're febrile and crabby, maybe vomiting, and so what do? You tap the shunt, maybe they're taken to the OR for a revision if there's a high suspicion, maybe if the ventricles have changed a tiny bit and there's no other explanation. And so what happens are these patients are getting, operated on, in some cases, repetitively, for a situation for which they should have never gotten a shunt in the first place. So when we're thinking about optimizing care for patients with hydrocephalus, even if we took the very meager goal of saying we could improve the situation by being more selective of who we shunt or who we treat from a neurosurgical point of view, and using genetic data to help make those decisions, I think has real value. So we're not even talking about gene editing or coming up with a drug to restore neurogenesis in the human brain. We're just talking about being increasing precision with how we make a diagnosis, so we can apply a more specific treatment that has a better chance of making the patient better. As you all know, these patients with and patient families of folks that have congenital hydrocephalus, this is a major problem for a family, right? Anytime your kid looks at you the wrong way or is a little crabby, you're thinking that this is potentially a shunt malfunction. You're calling the office, you're showing up in the emergency room. The rates of divorce in families with kids with congenital hydrocephalus, all types of hydrocephalus, is much higher than other population. This is a real problem, and so it's just something to keep in mind. When you look at the genes that are implicated in autism, cerebral palsy, microcephaly, other structural brain disorders, there is big molecular convergence between congenital hydrocephalus genes and other genes responsible for cortical malformation disorders. So it's totally fascinating to me that there, whether it's in schizophrenia, subtypes of autism spectrum disorder, the literature is rife with showing an increased ventricular size in these patients. And I think that what we're seeing here is just thinking about ventricular size as being a neuroradiographic biomarker of disrupted neurodevelopment or disrupted neurogenesis, early events driven by genetics impacting brain development with the final common pathway of ventricular size, and sorting out which of these patients with large brain ventricles need treatment versus not. I think genetics can be very illuminating. So this is just another gene that we and now others have found to be commonly mutated in hydrocephalus and other neurodevelopmental disorders. SMARC-C1, it's a member of the BAF chromatin remodeling complex, of which multiple components are mutated in a bunch of neurodevelopmental diseases that the neurologists take care of. But again, expressed very early neurodevelopmentally, neural stem cell regulator by impacting gene transcription. This is a pathway we all know, mutated in congenital, other congenital disorders, mutated in cancer, cortical malformations that go on to impact neuronal excitability and epilepsy syndromes. But a bunch of our patients have sporadic with sporadic congenital hydrocephalus have germline de novo mutations in the PI3 kinase pathway. So thinking about how does a germline mutation like that, in some cases, yield the hydrocephalic phenotype, right, without cortical malformations, without epilepsy, while in other patients, it may yield, an overgrowth syndrome, a brain overgrowth syndrome like hemimag. And I think that the answer is going to be a genetic modifier and the role of somatic variants in this context, being responsible for the phenotypic heterogeneity that we're seeing. And yet we can take these mutations that we find, and in this case, while P10 mutation that Tyrone Dispenza, an MD PhD student in the lab who will be graduating from PhD component in probably July, what he's finding is, again, a neural stem cell mechanism, but completely different from TRIM71. So whereas TRIM71 led to precocious differentiation and depletion of the neural stem cell pool and the neuroepithelium, leading to a highly compliant hypoplastic cortex and secondary, almost ex vacuo ventriculomegaly. What Tyrone sees is in activation of downstream mTOR signaling, leading to increased cell proliferation and growth that around the midbrain causes aqueductal obstruction and secondary obstructive hydrocephalus that can be rescued by administration of rapamycin to counter mTOR activation to pregnant mothers with pups. So just the concept that you can reverse or at least ameliorate a phenotype by targeting a genetic pathway in utero, for hydrocephalus is, I'm not saying we're going to be treating infants anytime soon by giving their mothers rapamycin during pregnancy, but as a proof of principle that this is a modulatable genetic event with a mechanism that is, in neural stem cells, I think is very fascinating, at least to me. So thinking about a neural stem cell paradigm of congenital hydrocephalus, this doesn't explain all cases, but it explains a significant percentage of them, and I think helps us think somewhat differently about using ventricular variation in ventricular size in humans as a unique pathway to learning about new pathways that are human specific, or if not specific, very important for human brain development. We think that given the number of cases that you can solve by just simply applying human whole exome sequencing to a sporadic congenital hydrocephalus patient that walks in your door, that we can, and given the cost effectiveness of that approach using whole exome sequencing, which you can now is $150 to do the entire exome of a patient. Thinking maybe about $400 for the proband and mom dad trio. We think that this is a potentially useful diagnostic adjunct in the diagnosis and even potentially treatment of congenital hydrocephalus. Down below is our maybe idealized version of what we think this platform is for congenital hydrocephalus, but also a platform that can be utilized and applied for other congenital neurological and certainly neurosurgical diseases where we have access to these patients and tissue. So the lab is not only interested in congenital hydrocephalus, but like I said, this is a platform that you can really apply to other maybe historically understudied conditions. And I think you guys don't see probably too many vena Galen malformations. Actually, not too many people do. It's a severe, yet probably still most common neonatal brain arteriovenous malformation that we found, mutations in FB4, RASA1, and other components of the this RAS signaling pathway. Dan Duran is now a University of Mississippi neurosurgical resident, highly talented guy who led that study. One study I thought you might be particularly interested in, just because it's so centered in the care of neurology, is trigeminal neuralgia. And so this hypothesis was something that really started with the observation that it's a disturbing observation, where as neurosurgeons, by the time these patients come to us after you've exhausted, carbamazepine and other, medical treatments, it's often suggested that, some of these patients, not an insignificant number, might have, vascular compression of the trigeminal nerve from a vascular loop or the, superior cerebellar artery or some other, vascular loop. But it's certainly the experience of neurosurgeons that even with, preoperative MRI that might suggest a vascular loop kissing the, the trigeminal nerve, that when you actually go in and look at the anatomy, often these are not in physical proximity, which suggests, wow, is this really an impact of vascular compression per se? And then, of course, there's a multitude of patients, and we all know them and maybe take care of them, who haven't responded to microvascular decompression. So, a retrosigmoid craniotomy, move the cerebellum out of the way, put a pledget between the vascular loop and the trigeminal nerve, and you realize that, wow, there's some patients who that don't respond to that treatment. What's up with that? So our hypothesis was that, maybe some of these patients, especially when they're younger, resistant to MVD, microvascular decompression, maybe they've had multiple microvascular decompressions. And certainly in those cases, which occurs with more frequency than you think, in which there are multiple family members, we thought that, maybe this has nothing to do about, vascular compression of the trigeminal nerve. And in fact, what we found is both transmitted and rare damaging de novo variation in genes highly enriched for GABA signaling. So components of the GABA A receptor itself, which is highly important in trigeminal system pathway, signal transduction. We were able to, in this paper, take a single missense mutation, and I think the gamma subunit of the type A GABA receptor, engineer it in a mouse, and recapitulate trigeminal neuralgia behaviors in mice with the single missense mutation in a gene that we found mutated in several human patients. And so we've applied this platform, or are applying it, I should say, in multiple other diseases. Arachnoid cyst is another one, where commonly thought of as, at least from a neurosurgical point of view, largely incidental lesions that don't need to be addressed, that aren't contributing to the disease, phenotype that you're seeing. But we all know, neurosurgeons, you guys as well, that there are multitudes of these patients that have other, sometimes subtle, associated neurodevelopmental phenotypes, like epilepsy, like autism, like maybe mild neurodevelopmental delay, neurocognitive delay. And from a neurosurgeon standpoint, despite some of the obnoxious looking scans that we see, we say, hey, if it's not causing hydrocephalus, this is not my problem and nothing to do. And in fact, what we found is that a large percentage of these patients, up to a quarter of them, have rare damaging de novo mutations, multiple recurrent hits in other OMIM disease genes that have been implicated in other neurodevelopmental disorders. So these patients, these arachnoid cyst patients, are highly enriched for these germline de novo variants in these critical genes that are important for brain development. And you can see how these break down, and you can come up with disease clusters in which certain gene variants are responsible for arachnoid cysts that localize to different, anatomical regions, whether it's the, middle fossa of, temporal fossa or versus P fossa, and that there are subgroups that just have more severe associated phenotypes. And so it's so what do you do with these patients? Well, as a neurosurgeon, sometimes these associated phenotypes are thought of as being caused by the arachnoid cyst or used for justification for doing a craniotomy for cyst fenestration to, for example, decrease the rate of epilepsy. Or we're thinking that if it's on the left side, it's somehow impairing language development or something, where in fact, what these cysts are really representing are they're essentially a radiographic harbinger that there is underlying neurodevelopmental pathology here that's due to germline disease mutation in a gene very important for early brain development. Usually it's going to be in a chromatin modifier that is highly expressed in the brain and is important for brain folding, cortical gyration. And really, you should not operate on these patients for that reason. And also, if they come to your office, you just shouldn't necessarily say, this is just an incidental finding, nothing to do, not contributing to the disease process, but say, oh, you know what, this signals that this patient should be, there should be early intervention or birth to 3, or get baseline neuropsychological testing so we can have data to look for impact of future treatments. So these patients who might often first get sent to neurosurgery because something is picked up on imaging, never get referred for a proper pediatric neurology workup, which you guys would do well. So future directions, there's a lot of them, whether it's applying the platform to different diseases or really pushing the computational pipeline with whole genome sequencing. I think we're just at the start of identifying somatic variation in the brain in different disease contexts. I'm fascinated about how aging might impact somatic variation in the brain, and also modify the phenotypes and uncover mechanisms of phenotypic heterogeneity for germline mutation. We've given a couple examples of mouse models that harbor disease causing mutations that we've identified in humans, but we have a bunch more that we think would be illuminating to uncover disease pathogenesis and maybe flesh out a drug target. From a clinical standpoint, I'm fascinated about making genotype, phenotype, radiologic correlations that can be fleshed out using machine learning algorithms, looking at large data cohorts, looking for patterns, radiographic patterns in which you could predict a gene mutation without actually doing whole exome sequencing. You could imagine how that would be very useful in the emergency room if you're trying to make a decision about whether or not to treat somebody or a choice of treatment. And then, fundamentally, as a clinician, just having more information to be able to meaningfully speak with family members, with mothers, families who are getting ready to deliver. I do all the maternal fetal care consults for pediatric neurosurgery at Mass General, in the MGB system, and so that's a lot of patients. And I'm just consistently appalled by the data that we use to prognosticate or to give information, to people, to mothers and fathers who are trying to make decisions about a pregnancy. I feel ill-equipped in doing that, and then recognizing also that you can have some incredibly severe radiographic findings on fetal MRI, massive ventriculomegaly, that really we know from a bunch of our patients, and having the genetics to sort out which cases of these they might fall under. Some of these patients with very large brain ventricles won't need to go on to need a shunt, or might have an IQ in the range of normal, that if just by looking at their MRI without that genetic knowledge, you might think that they're going to need a shunt, and the prognosis is poor. So just being able to have better information to help patients sort out or families sort out next steps in their medical care would be a very realistic, I think, future goal. So I wanted to thank the lab, our participating patients and families whose contribution to our studies, both in their enthusiasm, sending records, tracking down imaging, swabbing their cheek, etc., serves as the bedrock of all our studies. Both the Yale Rockefeller Centers for Genome Analysis, and now for the past few months, the Broad Institute up in Boston, which is not too shabby a place. These are all blessings and reasons for any of the success that we've had. So thank you for the opportunity, and if there are any questions, I'd be happy to take a few.
Dr. Kahle, this was an amazing lecture. We had a little time for questions, so let's see if there's any participants that want to take a question on the microphone.
Good morning. May I get the first question?
Sure.
I'm a senior pediatric neurologist, and I want to say thank you to you for a wonderful grand rounds. 3 things that I wanted to comment on was that, firstly, your very eloquent message to the students, trainees about passion and vocation. I think it's after my heart, so I thank you. And 2, of saying that this was, again, very illustrative of, again, as a clinician, what we have faced taking care of these children, and knowing that the difference between ventriculomegaly and hydrocephalus cannot be reiterated enough number of times. And thirdly, the wonderful advances we have made in being able to say, that all hydrocephalus is not the same. And in order to predict for a family what's going to be the outcome without being overly pessimistic or overly optimistic, of saying where our knowledge stands and where we are going. So I'll stop with these 3 comments, but I really, really, really thank you very much.
Well, that's very generous, and I think that those are, perhaps the main points. I could, I'll have to redo my summary slide with the notes that you just gave. And of course, they're not, they're very important, but they're also not revolutionary. These are insights that you, other neurologists, other neurosurgeons have been, anytime you take care of patients, that not all ventriculomegaly needs to be treated. Not all ventriculomegaly is hydrocephalus. That we need better information to communicate potential prognosis to patients, and having better data to do that. These are all no-brainers. In terms of technique, whole exome sequencing or approach, that's not new either. It's really just the I would say the dedicated focus and interest to apply it to some neglected conditions, I would say, or understudied might be a better term to use, and that there's still a lot to do. I think that maybe it's just sort of my antagonistic nature or skeptical nature of some things, but I just love thinking about some of these, at least in neurosurgery, and this would probably, confirm all the stereotypes that you have, but there are just a lot of dogma that people talk about very forcefully, and have done so for a long time. But yet, when you practice, and the longer that you practice, you see all the examples from your own experience that say, do you think that's the whole story? That's not my experience. Or, geez, there's a significant number of patients that don't fall into that model. What's up with that? And so, for example, the trigeminal neuralgia study, or even just the congenital hydrocephalus story. I have to admit that some of the genes that I found as a grad student were with Rick Lifton were implicated in ion transport and fluid transport, and one of the motivations, or I should say, a hypothesis I had was, my thinking was, I was like, oh, this is we're going to find a bunch of genes that are going to be these awesome choroid plexus, new transport genes or channels, and this is going to be a really coherent career for me, where I do find these genes for fluid reabsorption or ion homeostasis and all these different conditions that are now relevant for my own patients. And life will take you in directions that you were not expecting, and so the beauty of genetics is that it can do just that, where it can prove your own hypothesis wrong and put you in a zone where then you are, biology is always more interesting than your own ideas. And so it's been a real pleasure to, explore these pathways that didn't know anything about before. And to do that, of course, then that means you need to team up with people who maybe are an expert in that particular pathway for that gene. But it's always exciting and always new.
Okay, Dr. Kahle. I think we are just running out of time. I really thank you so much for all this wonderful lecture, all your time. I think there are many more questions that many of the audience have. I'm gonna just leave it we can contact you later, because I mean, this was a very fascinating lecture. I really know there are many more more participants. I want to just send you some questions that are very relevant for the clinical sometimes research here as well. Dr. Kahle, thank you so much for your time today. Really appreciate your time. Wonderful lecture again.
Thank you very much for the opportunity, and everybody stay warm this weekend with the snowstorm. I'll see you later.